PhD in microbiology and immunology. He was a Helen A. Whitney postdoctoral fellow and subsequently appointed assistant professor at Harvard Medical School in 2006, and later that year moved to Caltech. Dr. Mazmenian has won numerous awards, including a Searle Scholar Young Investigator of the Year at Harvard Medical School. Damon Runyon Innovation Award, was named by Discover magazine as one of the best brains in science under 40. Life Science Superstar by Genetic Engineering and Biotech News, and recently received the MacArthur Foundation Genius Award. His laboratory focuses on the study of beneficial bacterial molecules from the human gut microbiome as novel therapies for immunologic and neurologic disorders, with a specific focus on developing probiotic treatments for autism. He's a founder of 2 biotech companies and has or currently serves on the scientific advisory board of over a dozen companies, academic centers, and not-for-profit foundation. I would like to welcome Dr. Mazmenian. He's also the scientific advisory board of the Brain Foundation, and a great help to us. Thank you so much. Take it away.
Thank you, Pramila. While I still have you, I assume you can see my screen and hear me.
Yes.
And thank you for that wonderful introduction, and also for inviting me again this year to speak and participate in chairing this session. I'll pull up my slides. I really enjoyed the support from the Brain Foundation for our research, as well as the network that you're building of scientists for us to collaborate and really think about pushing the scientific boundaries of this really obviously interesting scientific area. So I'll chair this morning's session, which is loosely themed basic and translational research in ASD. And I'll start by discussing some of our work, give an introduction to a project, and then the second half, talk about some newer work that is being funded by the Brain Foundation. And almost everything I'll present today is unpublished. And so obviously the microbiome is familiar to the audience and was discussed yesterday, but just roughly speaking, we have 100 trillion bacteria living in and on our tissues throughout our entire lives. They contribute to the genetic material in our body. In fact, many people have termed the microbiome the second genome, because there are actually 150 times as many gene families in our microbiome, which is the collective genomes of our gut bacteria, than it is in our own genome. And so the 2 genomes essentially are working together to mediate the balance between health and disease. And the bacteria themselves have the metabolic capacity in aggregate of the liver, and weigh approximately the same as a brain. So it's this foreign organ that we all harbor in our bodies that we believe critically contributes to our biology and to our health. And it's been shown, and I'll just go quickly over the disease areas. It's been shown that the microbiome is related to a number of different disorders and conditions, many of which have been done in animal models. And I think that that's still where the field is, that though a lot of work is being translated, and again, we heard some of this yesterday, the vast majority of the discoveries and the innovation that is coming in the field still happens in animal models. I think that's reflective of, again, the fact that this is a new research area, but also, there are still tools that are being developed and other technologies and even regulatory aspects that are being put in place for an increase in human research in the microbiome. I think we need that, and there's more coming. Just loosely in terms of how the microbiome can interact with the brain, there are a number of different conduits that include the production of neurotransmitters in the gut under the regulation of microbes, use of the immune system, where most of the immune cells in our bodies are in our gastrointestinal tract at any one given point in time, and they're educated or primed by gut bacteria, and then these immune cells or their products, such as cytokines, can circulate throughout the body and even have been shown in animal models to mediate behavior. Neurons can have receptors for these immune cytokines. What I'll talk about today is this class of communication where molecules directly produced by bacteria can enter the circulation, in some cases get into the brains of animals and modulate physiology, brain activity, and behavior. And then there are obviously neuronal connections as well that link the gut and the brain. And it's really important to remember that this is there's a lot of evidence that the communication is bidirectional. There's signals going from the gut to the brain, and then from the brain to the gut. We can all think about this in our own lives and day-to-day occurrences of how maybe when you're stressed in terms of your gastrointestinal tract, then it may, you may have difficulty in thinking or sleeping, or if you have anxiety or depression, something's on your mind, it may affect your GI tract. Again, those are just broad examples of the connection between these two organs. But experimentally, there's still a lot more work that's been done in looking at gut to brain connections, just because it's technically easier. And I would argue we still need many more studies where the trigger or the inciting signal is in the brain, and we need to understand what's happening in the gut. But again, it's bidirectional in communication. But today we'll focus on molecular signals that emanate from the intestines and affect brain function. And so a few years ago, I won't give you the background for this paper. Obviously, it's been published quite some time ago, but we showed in this manuscript that the microbiome is altered in a mouse model of neurodevelopment. This is the maternal immune activation, or the MIA model of neurodevelopment, which has many features of autism and schizophrenia. And in addition to an alteration in the microbiome that was coupled with behavioral changes, we found changes in the gastrointestinal tract, both in terms of epithelial barrier dysfunction as well as inflammation. And then wanted to understand if there's a link between the gut and the brain, just as I described in this previous slide, and then performed an untargeted metabolomics analysis. And what that essentially means is we looked for small molecule differences between mice that had behavioral symptoms consistent with autism and mice that did not. And so again, I know I'm not providing all the background for this, but because I want to get into the unpublished work. But very briefly, these are animals that do not show features of autism. These are animals that do show features of autism, and I'm talking specifically about behavioral features. And you can see that in the animals that have behaviors that are autism-like, that there's an elevation of this metabolite called 4-ethylphenyl sulfate, or 4-EPS. There are other molecules that were dysregulated, but we focused on 4-EPS, almost a 40-fold increase in this one metabolite, which was the highest change in the entire data set. And so over the past few years, we've been trying to understand what the function of 4-ethylphenyl sulfate is. And what we suspected, and we now know, and I'll show you data for this in a minute, is that bacteria can convert dietary tyrosine, excuse me, to a number of different molecules, one of which is 4-ethylphenyl. This happens in the microbiome, this happens in the intestines. And then when 4-ethylphenyl enters into the circulation, we believe it's very rapidly sulfated to 4-ethylphenyl sulfate, presumably in the liver, though this reaction can happen elsewhere. And then ultimately, the molecule that is in circulation and even in the brains is a metabolite called 4-EPS, the sulfated version of 4-EP. And I also want to emphasize that though I'll talk exclusively about the biology of 4-EPS today, we really think that this is a harbinger of information for additional metabolites. There are many dozens of metabolites that fall into the same category. They're either tyrosine or tryptophan metabolites, but they're these phenolic molecules produced by the microbiome, which in many cases seem to co-vary with each other, meaning that they go up and down in the same individual, and presumably have biological functions that are either similar, overlapping, synergistic, and need to be studied. So again, I'll talk about one metabolite, but there's many, many more metabolites and combinations and reactions that we believe are happening in vivo. And so, as I just presented, we discovered this elevation of 4-ethylphenyl sulfate in this animal model, in a mouse model of neurodevelopment. And the reason why we discovered it was because it was in a particular panel of chemical standards for an untargeted metabolomics analysis done by mass spec. And what had happened is that there had been previous to this a number of different metabolomics studies in humans, but they just weren't picking up this molecule. It may have been in the biological tissue, whether it's urine, plasma, or the tissue that we looked at, but it had not been reported. And so we went back and addressed this issue. So working with Paul Ashwood, at UC Davis were able to obtain samples from a charge study that had run there many years ago, where they had collected plasma and feces from a large number of individuals with autism and typically developing individuals. And so this was obviously interesting to us because very, very few studies, even to date, have collected feces from individuals with ASD. And so we did a metabolomics analysis. There are many, many metabolites. There were changes to the paper we published last year, but I'll just draw your attention to 4-EPS. There's a subset of individuals between 30 and 35% of the ASD population, at least in this cohort, which had these very, very high levels of 4-EPS compared to individuals that were in the typically developing group, or samples from individuals in the typically developing group. So it's not all individuals with ASD. I mean, the metabolite is detectable in all individuals in the study, but then there's this subset where the elevation of this metabolite is quite high. And then if we think about what smaller subset of individuals show the greatest increase or fold increase in 4-EPS, it's in those individuals that have an ASD diagnosis with GI issues. And so individuals with ASD without GI issues have about 5.5 to 6-fold increase. Those individuals with GI issues have over 9-fold increase. So again, more suggestive that there may be issues related to the gastrointestinal tract in this metabolite. And so we wanted to, again, understand the function of this metabolite and go beyond these correlations. Of course, this metabolite and or other metabolites can serve as potential biomarkers for disease progression or for potentially even interventions. But we want to understand, is there a physiological or biological function to these metabolites? And so obviously we start as typical in biomedicine, we start with animal studies, because these are proof of concept studies to just convince us or to demonstrate that there are indeed physiological changes relative to this metabolite. And so we've done a number of experiments where we've fed the metabolite, we've injected the metabolite. I won't talk about that data, but it's all consistent with what I'll show you. But working with Michael Fischbach at Stanford and his very talented postdoc Masa, who essentially deconstructed the biosynthetic pathway for conversion of tyrosine to 4-ethylphenol, which I just alluded to. And so what Michael and his team discovered was that there are 3 enzymes, all bacterial enzymes, that can convert tyrosine to paracoumaric acid, which is then converted to 4-vinylphenol and then ultimately to 4-ethylphenol. Again, this all happens in the gastrointestinal tract. The system can use paracoumaric acid or picoumaric acid as a substrate as well. That's found in certain dietary compounds in certain plant-based foods, but the system appears to prefer to use tyrosine, which, as many of you know, is an amino acid that's found in diet, and also we get from our bacteria as well. And then what Michael did was that he engineered bacteria to either produce or not produce 4-ethylphenol. So the exact same 2 strains of bacteria, Bacteria ovatus and Lactobacillus plantarum, that either do not have the genes for 4-ethylphenol production or do have those genes. And so if they do have the genes, these are 4-EP plus bacteria, and then we call the same exact organisms, just missing 3 genes, the 4-EP negative bacteria. And so again, 2 bacterial strains for each pair that are different, to the best of our knowledge, in the production of this final metabolite 4-EP. And so we colonized notobiotic animals with the 2 strain pairs or 2 sets of bacteria to produce mice that either do not or do produce 4-ethylphenol sulfate. And again, this is a reductionist system, but it's a very clean system in the sense that we know that all the biological activity that we observe in this animal is dependent on the production of 4-ethylphenol in the gut. And so we show that to be the case, is that these bacteria do produce 4-EP in the feces if they contain the genes for conversion of tyrosine to 4-ethylphenol. When we look in the urine, and we see the same data in the blood, that there's a high level of 4-EPS in the circulation. And very interestingly, we can detect almost no 4-EP. So it's a very quantitative conversion of 4-ethylphenol to 4-ethylphenol sulfate. But again, we can see levels of this metabolite in the circulation. And we can also detect 4-ethylphenol sulfate in the brains of these mice if the bacteria can produce 4-EP in the gut. And so the metabolite reaches the brain. We don't know if the final function is in the brain, but it's interesting that the metabolite is indeed there. And so we did a number of studies I won't talk about for the sake of time, where we looked at brain activity and brain connectivity, and we see some very interesting changes in particular regions of the brain that are more or less activated in the animals that were exposed to 4-EPS. And then we wanted to understand if we can explain those brain-wide changes that I just described at the molecular cellular level. And so working with Dan Geschwind at UCLA, we performed an RNA-seq experiment for several different brain regions in each of the 2 cohorts of mice. And almost all of the genes, or the vast majority of the genes, over 75% of them, that were dysregulated either up or down at the transcriptional level on the paraventricular nucleus of the thalamus, or the PVT. And you can see that that's here in green. Many of the genes are found in neurons. So when we do cell-specific enrichment for these transcriptional pathways, we can see that there's certainly changes in the neurons, but also it looks like there are changes in these cells that are various different maturation states of oligodendrocytes. So OPCs are oligodendrocyte precursor cells, NFOs are newly formed oligodendrocytes, and then MO is mature oligodendrocytes. I'll talk about oligodendrocyte biology in just one second, but we wanted to validate to see if indeed the genetic signature that was being picked up by the RNA-seq was giving us the insight into oligodendrocyte biology. And so there are a number of genes associated with oligodendrocyte precursor cells, and these genes in green are all elevated in the 4-EP positive mice. There are a number of genes associated with mature oligodendrocytes. All of these genes are reduced in their expression level in the 4-EP positive mice. And so this really gives us this picture that there is, it appears at the genetic level that, or the transcriptional level, that there's this arrest, if you will, in maturation of oligodendrocytes. They seem to be stuck in this OPC state, generally speaking, and reduced in their efficiency to convert to mature oligodendrocytes. At least that's what the RNA data told us. And so we wanted to now validate that to be the case. So using immunohistochemistry for particular markers that can differentiate mature and immature oligodendrocytes, we showed that indeed the mice exposed to 4-EP, so the animals that had 4-EPS in their brains, have less mature oligodendrocytes when we use, again, this marker of mature oligodendrocytes CC1 over NG2, which is a marker of oligodendrocyte precursor cells. We've used a number of other markers as well, again, not shown here for the interest of time. But importantly, the number of oligodendrocytes in total is not different between the two. So we don't think there's an issue in terms of development of oligodendrocytes, but some likely dynamic delay or arrest in their maturation. And this is again by immunohistochemistry. We can validate this by flow cytometry as well. So these are the mature oligodendrocytes that are found in abundance in the 4-EP negative mice, and they seem to be missing in the 4-EP positive animals. And so again, this is quantitated here. So the function of, so again, all the data suggests that there is either an arrest or some sort of halt in the maturation of oligodendrocytes in the brains of these animals. It is region specific. It is certainly found in the PVT and other regions as well. And looking at the subsequent effects of this delay in maturation, again, as many of you may know, oligodendrocytes have a number of functions, primary to which are the laying down of myelin around neuronal axons. And so myelin helps conduct signals across axons over long distances. So think of this as like the coding on a copper wire in your home. And there's increasing evidence that certain behaviors and different neurodevelopmental conditions are associated with changes in myelination patterns in the brain. And so having seen this effect at the cellular and transcriptional level in oligodendrocyte maturation, we wanted to see, is there a change in myelination in these animals? And so initially looking at the corpus callosum, where the neuronal fibers are aligned, so it's very easy by electron tomography, which is what we're looking at here, to physically quantitate the degree of myelination as well as the thickness of myelin. We can see that there are many more unmyelinated axons in the 4-EP positive mice compared to the 4-EP negative mice. And when we do analysis called a G ratio, which takes into account the variable shapes of these cells, we can see that the higher G ratio, which correlates to thinner myelin, is found in the 4EP positive mice. And then when we actually just look at the thickness of myelin in these tomograms, we can see that the myelin is thinner in the mice exposed to 4-EPS. So again, consistent with the arrest in oligodendrocyte maturation. And then more specifically in the PBT, here we used functional ultrasound to look at the organization of myelin in the structure. We looked brain-wide as well, and we see very similar effects, that there's just more disorganized myelin in the 4EP positive mice compared to the 4EP negative mice in this particular region, again, where we saw that strong genetic signal. And then ultimately, we wanted to now correlate this back to behavior and ask, is 4EPS altering behavior in these animals? So we performed a wide variety of behavioral tests. Here I'll show you the open field test, where animals are put into an arena, and the more time they spend against the walls, the more anxious those animals are, the more they venture into the center, the more bold those animals are. And the animals exposed to 4EP in the gut, which again have 4EPS in their circulation in the brain, spend less time in the center, more time at the wall, suggesting that they have an anxiety-like phenotype. There's no change in locomotion, which is an important control, because if the animals were sick for any reason, it would affect this outcome. In fact, we've done an extensive amount of analysis on the health of these animals. We see no health differences in any parameter. We've looked at GI health, brain health, weight, immunologic inflammation, and a number of other parameters, and really can't find any signs of overt disease or illness in these animals. They seem to be perfectly fine, but they seem to choose not to enter the center. And this is supported by data from the elevated plus maze, another test for anxiety, where animals that spend time in this open arm are just less anxious and more bold, and the more timid animal will spend time in the closed arms. And you can see that the mice exposed to 4-EPS should spend less time at the terminus of the open arms compared to the 4-EP negative group. We've actually shown in 2 additional tests that these mice have anxiety. So that's a very, very strong phenotype. Excuse me. But we also wanted to look at Asd-like behaviors. So we looked at social interaction in the direct social interaction test. There is a non-significant decrease in social interaction, which is consistent with social deficits, in this peculiar change in anogenital sniffing, which I'm not sure we can fully understand, but is a known behavior of animals. When we looked at ultrasonic vocalization, essentially how these animals communicate with each other, there's a decrease in the duration of each call. So every time they emit a call, their sentences are shorter, if you will, as an analogy. So these animals appear to have emotional changes in emotional behaviors that are consistent with anxiety and with autism. And we did motor test these animals. We found no motor deficits. We tested the animals for cognitive learning and memory tests. We found no changes between the 2 groups. So it doesn't look like there's a gross change in behavior in these animals. It seems to be more confined to emotional behaviors. And so just to summarize, this project and this preclinical work, either changes in the gut or the products of the gut, known as the metabolome, small molecule products known as the metabolome, can be different, either in animals and potentially in humans. And then this leads to a change in the flux of microbial metabolites across the gut barrier. And in the case of 4EP, 4EP is elevated and can then be sulfated in the liver, and essentially its concentrations increase in the circulation, and the metabolite does enter the brain, and it leads to an arrest in oligodendrocyte maturation, which then subsequently leads to a reduction in myelination, both in terms of the number of axons myelinated and the thickness of the myelin itself. And then that alters behavior. We have some functional data that does correlate the myelination and the oligodendrocyte phenotypes to the behavior. So we really think it's working through oligodendrocytes. And so, so this work, obviously, we're interested in identifying what are the receptors for 4-EPS, what are the specific cell types that are being directly impacted by this metabolite, and very importantly, to learn the function of the many other metabolites as I mentioned, that we believe are in this same chemical class and may have biological functions as well. And so Stuart Campbell later this afternoon will give a talk on how this work has been translated into the clinic. So again, we run a basic science laboratory, but Axial Therapeutics, where Stu is the CEO, has taken this technology into the clinic. And again, I urge you to listen to this talk. And so we, all the work that I just showed you has been done in wild type animals, but of course, autism is a genetic disorder, and we want to then understand what are the effects of either the gut or the microbiome in the context of ASD-related genetics. And so we started working with the Shank3 model. And so what Shank3 is it's a gene that's believed to be the cause of Phelan-McDermott syndrome, whether it's through deletions in 22q13 or mutations that lead to insufficiency leads to Phelan-McDermott. And most individuals with PMS have ASD. I think over 70% of Phelan-McDermott individuals have autism spectrum disorder. And then they have a range of different behavioral and physiological features, which do include gastrointestinal and elevation of gastrointestinal issues. What Shank3 is, it's a scaffolding protein in the postsynaptic density of excitatory neurons, and it has an effect in glutamate signaling. It's been widely studied. Again, I'm sure many of you know the literature on Shank3, and of course, Joseph Buxbaum is an expert in this area, and he'll be speaking next. And so again, we wanted to see if there were any microbiome or gastrointestinal effects in the Shank3 model. And this is there is a mouse model that had been developed several years ago, where it's a deletion of Shank3, which results in several behavioral changes in the animals, this decrease in rearing in the open field. These animals groom themselves excessively. So here you can see the knockout animals, Shank3 knockout animals, excessively groomed to the point where they have these lesions, and then they have an anxiety-like phenotype as well. And so there's a well-established mouse model. There are even non-human primate models for Shank3. And it had already been shown that the microbiome is related to the Shank3 animal, in the sense that when people had compared wild type to Shank3 mice, that there's a decrease in Prevotella and Lactobacillus species, and then supplementing Shank3 animals with a particular organism, Lactobacillus reuteri, changes their behavior in the social paradigm in 3-chamber social test, as well as changes in the receptor for GABA, in particular brain regions. And so again, there's emerging evidence the microbiome plays some role in the physiology and the behaviors of this metabolite. There's an unpublished, I believe still yet unpublished bioarchive paper from Drew Correlate at Mount Sinai, who Joseph works with, that has shown that antibiotic treatment appears to exacerbate symptoms in this mouse model. So just very quickly, these are the knockout animals with antibiotics, and they spend less time with another animal compared to an inanimate object. So they show the social defect, which is exacerbated over the animals that weren't treated with antibiotics, and that's quantitated here. And then same in the open field, there's an anxiety phenotype that is increased upon antibiotic treatment of the knockout animals. And there are also changes in gene expression in this paper as well. And again, here's the reference for that work. So there's preceding evidence that the microbiome is both altered and appears to impact behaviors and other outcomes in this animal model. And so Jessica Griffiths in our lab is interested in what's going on in the gastrointestinal tract of these mice, and has performed a whole gut transit assay, where she essentially feeds a dye to the animals and looks at how long the animals take for them to pass this tracer, if you will. And the gut transit time, in terms of the whole gut, is much faster in the Shank3 animals compared to the wild type animals. So essentially, there's a faster movement of contents through the entire gastrointestinal tract in the Shank3 knockout mice. And when she did an experiment where she isolated the effects of just the small intestine, she wasn't able to see a different. So there's a different diet and a different protocol to ask, is there an effect on gastric emptying or in small intestinal transit? And she does not see a phenotype there that would explain that. So what we think is happening is that there may be a change in the colon that results in this effect. And so what Jess did was she set up an ex vivo system where we can take colons of animals and put them in this organ bath, and then using a video tracking software, look at the contraction of the muscles in the colon and ask, are there differences between mice, between the genotypes, is what she's done here. But the system also allows us to flow neurotransmitters, other metabolites and molecules through the system to see if gut motility has changed. And so in this CMMC assay, and this is live tissue, which you can see here, the data show when there are contractions based on the width of the tissue. When there's contractions there, the gut tissue is narrower, and you can see that in the red. And indeed, in the colons of the Shank knockout animal, there are more migrating complexes, so more muscle contractions, if you will, in the knockouts compared to the wild type, which likely can explain the whole gut transit data that I showed you previously. So we're obviously very interested in why knockout or deletion of SHANK3 in the colon appears to result in this change in contractility. We're following that up. I should mention we have animals where we can selectively knock out or express SHANK3 in the gut and the brain. This is also funded by the Brain Foundation grant. And we wanted to understand, are there physiological changes if we just have SHANK3 in the brain or just have SHANK3 in the gut, and how that affects either GI or behavioral functions. And those are ongoing studies. What just wanted to then understand is there now a susceptibility in the colons of these animals to insult? And so we used essentially a colitis model, the dextrans sodium sulfate model, DSS model, to ask, are there changes in essentially inflammation or colitis. And initially you can see that the Shank knockout animals are more susceptible to DSS colitis. They lose weight compared to the wild type animals. And then when we look at the colon, they have this hallmark shortening of the colon, which is consistent with not just mouse models of colitis, but of human ulcerative colitis or Crohn's disease as well. And so essentially the intestines of these mice are just more vulnerable than the wild type mice. And we've also become quite interested in the microbiomes of these animals. So I showed you a few slides ago, there were 2 studies that have shown that the microbiome is different, but we know the microbiome is different for animal models in general, because commercial mice just have very depleted or reduced microbiomes. And so this is actually reflected here from this recent paper. These are the microbiome profiles of one of the 3 large commercial vendors. And as you can see, these microbiomes really are diminished or reduced in the diversity of the microbes that they harbor. And then these are microbiomes from animals, wild mice caught in 3 different locations. This happens to be in Maryland. And you can see how feral or wild animals just have much more diverse microbiomes. And what's really interesting is if you take the same laboratory strain of mice and then colonize them with either wild microbiome and compare them to the commercial microbiomes, you see a very dramatic difference in the immune response. I mean, it's pretty remarkable to what degree the immune system of these animals are shaped by the diversity of the microbiomes in them. And this has been done for a number of other papers, but clearly you can see here that the wild mouse microbiome just has a higher pro-inflammatory response than that of the laboratory mice. So I want to see if this principle applied to neuroscience. So we took both the wild type and the SHANK3 knockouts, and then we essentially diversified their microbiomes by adding these wild mouse microbiomes. And what you can see, some really interesting features, is that just in wild type animals, there's this increase in I should explain the behavior test. This is the O maze, zero maze, where it's very similar to the elevated plus maze, but instead of being a plus, it's a 0. And so here are the animals where the animals feel safer because it's in this closed arena. Here's the open arena where the animals are less anxious if they spend time there. And clearly the animals that were colonized with the wild microbiomes are less anxious. And we see this really interesting, reveals this really interesting genotype effect in the wild R microbiomes if the Shank3 protein is knocked out. And so this is in males, and we can see less of an effect in both sexes, but clearly the wild microbiome is changing the behaviors of both the wild type mice as well as the knockout animals. And that's true in the open field as well, but here the effect is more pronounced in males and females, where you can see that the animals that have the wild microbiome that were knocked out in Shank3 spend more time at the walls, which is where they feel safer, meaning they're more anxious compared to the wild type mice. So obviously more studies coming, and we wanted to understand how this affects other behaviors, how this affects GI function. But I think that there is a lot of biology that we're missing in the way that we house and rear laboratory mice, and some of that is revealed by just diversifying their microbiomes to be more representative of what an animal would look like in nature. And so I'll just leave you with this concept, just at 30,000 feet of the balance between genetics and environment in studying autism. And I think oftentimes the role of environment and the role of genetics are studied separately. In other words, people look for genetic etiologies, other groups, other laboratories, other teams look at environmental etiologies. I think there's just a lot more work that needs to be done at the interface to look at gene-environment interactions and to see to what degree those may explain outcomes in animal models, and certainly, most importantly, in people. And I'll just finish by thanking Brittany Needham, who did all the work I showed you on for EPS, and Jessica Griffiths, who's a graduate student in our laboratory, who led the Shank3 project. These are all of our collaborators on the work that we do in our laboratory. Here are our funding agencies, and huge thanks to the Brain Foundation. So I'll stop there, and I think I'm over a little bit, or quite a lot.
Thank you so much.
But I will stop there, and I will ask Pramila to maybe moderate this session. And here we go, moderate the session to see if there are any questions in the chat, the Q&A.
Yeah, there's one question from Dr. Quadros. Is the gut distribution of bacterial effects dictated by region, such as ileum versus colon? And there's another question, does maternal gut microbiome affect the brain and subsequent behavior?
So the first question, we don't know about the regional effects in the small intestine. At least the transit time data suggested that the defect or the change is not in the small intestine, so we haven't looked carefully, and so we just don't have that data. And so we're focused a little bit more on the colon. And again, it's a new project, so there's still a lot of exploring we need to do. But the colitis data also suggests that there's a compromise in the colon. So I think that's where we'll focus. The maternal microbiome of all animals, it is important, meaning that the microbiome of the mother is passed on to the offspring at birth, and that's the first exposure to live microbes and shapes the microbiome. There's a number of studies from many groups. I think Jun Hu, who spoke last year at this meeting at Harvard Medical School, has been doing some of the most pioneering work in the space in the past few years, I think, where he's shown that through various different inflammatory effects, the maternal microbiome affects behaviors and GI outcomes. He had a paper just last week on this topic. But of course, we and many others have worked on the effects of the maternal microbiome, and it appears again in animal models to be important in shaping both behaviors, GI function, as well as inflammation.
One more question. What is the age range of the mice in your study, and how does it correlate to human age? And do you look at changes longitudinally?
The age range of these animals, technically speaking, is we behavior test the animals at 7 or 8 weeks of age, which is adolescent, I think roughly correlate to adolescence in humans. And we are not trying to model the age component, though we can, and obviously it's an important variable in all of our experiments. We're using time points, that have been established for both the Shank3 mouse as well as just various different behavior testing paradigms in all animals, and they show the effect there. But we haven't looked at earlier stages, which can be a challenge because animals have to develop to a certain point before they show these complex behaviors. So unless the animal is socializing, you don't know if there are changes in socialization until they're vocalizing. You don't know there are changes in vocalization. So there is a lower age range where we can't test the animals, but we haven't gone to the older animals. We haven't taken this particular paradigm to mice that were anything older than about 10 or 12 weeks of age. So we're still looking at very early stages of the animal's life.
Thank you very much. I think that's it for questions. So should we move on to the next talk?
We shall. Thank you very much, Pamela. And so I'll introduce the next speaker. The next speaker is Joseph Buxbaum. And the title of Joseph's talk is Genetics and Genomics at the Mount Sinai School of Medicine. And again, I'll introduce Joseph. He'll start the talk, and then he is going to introduce Matthew Lawley after him.
Thank you, Sarkis. Let me just get to the screen sharing part. All right, I'm going to just do one thing. Where is annotate? Don't see it. All right, I'll start. But so thanks for this great opportunity. Thank you, Sarkis, for the introduction, and thank you, Pramila, for inviting us and for supporting this work. So this is slide 2. Let me go back to slide 1. Oops, it's going the wrong way. So basically, the project that we're funded to do is connecting rare genetic mutations in autism to common biological pathways. And Matt Lally and I will kind of share the responsibility. I'll talk about the past, what we know about rare genetic mutations. And then Matt will talk about his efforts and our efforts to put these rare genetic mutations into common biological pathways. This is supported by the Brain Foundation. We do not yet have data, but Matt and team are moving very quickly, and we're actually on schedule to finish everything as proposed. So just taking kind of a long view of rare genetic mutations in autism, I'll kind of highlight the work of the Autism Sequencing Consortium, which really started with the last stimulus package in 2009, the American Recovery and Reinvestment Act by these two gentlemen here who signed it off. One retired and one came out of retirement. And a small amount of that stimulus money went to the NIH for some crazy ideas. And one of the crazy ideas was whole exome sequencing, which was a technology that, or whole genome sequencing, that was now really becoming available academically within reasonable prices. This was the first unofficial or official meeting of the ASC in 2010 in New York City. We brought a lot of the funders and main labs who were doing sequencing together. And what was going on in 2010, the papers came out in 2012, was whole exome sequencing was supported by the ARA Act, and several labs, 5 labs, decided to work together with ARA funding and sequence, a huge number of samples in those days, 50 trios a laboratory, and put all the data together and see what we saw. At the same time, the Simons Foundation was doing something pretty similar, funding 3 laboratories to work on the Simons SSC samples. And so over the course of 2012, there were these 4 papers had about 1,000 ASD families and focused on de novo or recent mutations in autism. And collectively, if you analyze it jointly, there were 9 genes that were shown to be contributing to autism in a very significant way. By which I mean, if somebody had a mutation in that gene, it would probably contribute to most of their developmental phenotypes. And based on that, something called the hidden species problem, a simpler version is the birthday problem. But basically, you could estimate with 20,000 genes in the genome, how likely is it that you're going to find 9 genes currently hit by mutations out of the greater number of autism genes that should be in the genome. And at that time, in 2012, the data was pretty consistent and hasn't really changed, that about 1,000 genes in the human genome, 1,000 out of 20,000, can be changed in such a way to contribute to autism risk. So in a certain sense, we all have autism variants, and because 5% of our genes are directly involved in some of the processes that underlie the social manifestations of typical development and atypical development. So the argument basically, and just to make it clear, if there's 1,000 genes, right, and we had finished 1,000 cases in 2012, we're far away from discovering all the genes that we can, right? We're at the very early stages. And so the argument of the ASC was, let's put together all of our data prospectively, not publishing small findings at a time, but try and get all the samples run concurrently, which was a bit of a stretch in those days. It was the first example of its kind, really, of large-scale prospective data sharing and sample sharing. The first paper came out in 2014 and now had 22 genes that were autism genes, overlapping with many other developmental disorders. Shank3 you just heard about. I won't talk much more about that, but that's a good example of a gene where the mutation contributes to the phenotype: autism, intellectual disability, developmental delay, epilepsy, and obviously also GI abnormalities. The most recent paper from the ASC, from the Autism Sequencing Consortium, came out last year, and now it has 102 genes all across the genome, and there's Shank3 again, that each one, when mutated in that individual, contributes enormously to the risk for autism. And what's gone on to bioRxiv, so this is kind of what we look at. The total sample size is in the tens of thousands of samples and 12,000 cases in individuals with autism. And each one of these funny-looking conglomerate of letters is the name of a gene. Of the 20,000 genes of the genome, here are 25 or so that, a fraction of individuals with autism have mutations in that gene. And here's another group of genes where a smaller fraction have mutations in that gene. There's Shank3, which you just heard about. And so, obviously, if you are working on Shank3 as an example, you have a biomarker, for lack of a better word, where you can find people that have a mutation in Shank3 and study them directly. And you can make a mouse model, as Sarkis told you, that has a mutation in Shank3 and understand what's going on in that mouse, which reflects what's probably going on in patients with that mutation. So you can take basically a rare disorder or orphan disorder approach to these genes and say, I'm not studying autism, or I am studying autism, but I'm studying the autism that's caused by mutations in Shank3. The most recent analysis by the ASC now doubles the number of cases, 20,000 cases, more from the ASC, but also many more from the Simons Foundation. And we now have 71 genes at a very, very strict threshold. But at a kind of the typical threshold we tend to use, we now have 250 genes that contribute to autism risk. So remember I said there's about 1,000. So, we're honing in on 25%, which is pretty impressive. And this is the genes listed by basically by evidence of their association. There's Shank3 you just heard about. ADMP is one of the genes. We study Shank3, ADMP, FOXP1, a couple other ones. So you can select these genes here and approach them, as I said, as a rare disorder. This study really was, I think, another important step in gene discovery in autism. And it's really been led by a conglomerate of individuals who've done an amazing job of bringing the SPARC data and the ASC data together, and also bringing in other kinds of variation, including copy number variation in the analysis. So that should be online. I think it was submitted to bioRxiv on Friday. It should come live in just a couple of days. So, when I started this business of autism discovery, gene discovery, we thought maybe there'll be a couple of genes. And I remember very well when the paper came out and said that there are going to be 4 or 5 genes underlying autism based on how it looks in families. And the idea was that if we could find the gene, the autism gene, or even those 4 or 5, they would highlight a single pathway, right, out of many one, right? There'd be 4 or 5 genes, strong genes. There'd be one pathway, and we'd know where to go for therapeutics. I can we can talk about this at length, but first of all, we've gone from 4 genes to now 1,000. And every time we look at those genes, there's clearly not one pathway, right? There are neurons, and Sarkis has talked about some of the indirect effects of a neuronal gene on oligodendrocytes. There's lots of ways where you can disrupt the brain to cause behavioral abnormalities of various kinds, including deficits in social behaviors and the presence of repetitive behaviors. So, what I've already kind of intimated is that a lot of groups scientifically are focusing now on a gene by itself. So out of many, E pluribus multis, right? So instead of saying we treat autism as an entity, we could treat autism as a series of independent disorders, right? So your therapeutic focus is on gene-targeted approaches. You think about the treatments that are going to help somebody with a Shank 3 mutation or Mcdermott syndrome. And it may or may not help the next person over. It may, but it also may not. So it's kind of what we do in cancer now, right? And, this is from an epidemiological study in Sweden. If you just take current genetic knowledge, you can actually make a genetic finding in about 28% of individuals with autism. So, there is a path forward for, at least this slice of the pie to say, hey, we can take a rare genetics or orphan disease approach to autism. Of course, there's another larger group that has no genetic diagnosis. So we will make more genetic diagnoses as the number of genes increases. But there's also going to be a significant number that are not going to have a formal mutation that explains much of the phenotype. So this is very kind of where biotech and certainly academia is in autism. They're thinking about a lot of these genes like Shank3, ADMP, POGZ, SNC2A as rare diseases that can be kind of approached with the orphan drug perspective. And there's a lot of incentives to orphan drug development. It is probably one of the most robust areas of therapeutic growth right now, at least in higher income countries. Some of it has been gene therapy. Some of it's been just remarkable just in the past couple of years. Kids who have certain kinds of developmental abnormalities that will end in their very early death can now get a single injection in the spinal cord and actually, so far, certainly be doing very, very well, right? So the idea of gene therapy or gene-targeted therapy has really come into its own when people are realizing that it can make profound differences in developmental disorders. Obviously, these ones that we're talking about are more severe than autism, but if the treatment or the intervention is safe and the individual has a very difficult trajectory, no language, severe anxiety, severe attentional deficits, low IQ, you begin to think about interventions to kind of to reduce some of those disabilities. Right? So I mentioned that we take this approach of rare variation in autism, and we do a pretty deep dive on 5 genes, Shank3, ADMP, FOXP1, DDX, which is on the X chromosome, and CHAMP1, which actually comes from another study. We won't go into this, but CHAMP1, ADMP, some of the others actually do form a complex and act together, but others do not, right? And what we try and do with each of those genes is we try and do in vitro work. We try and work on iPSCs or stem cells. We try and look at genetically modified rodents, and we also see patients. So Sarkis was nice enough to talk about some of the genetically modified rodents of Shank3. We've made a couple, not the one Sarkis talked about, but he mentioned the work of Drew Correlate, which is one of the mice that we've made. But we've also seen a lot of patients with this mutation, and a lot of the phenotype, not only the autism phenotype, but the other behavioral and other phenotypes in Infilo-Mcdermott syndrome have been carried out by groups like ours that bring in these individuals who really aren't known medically until you bring in a good number of them. And that you also learn something about endpoints for clinical trials. You can develop patient biomarkers, you can look at the mice, and you can do stem cell work. And so this idea that maybe, if you look at the mice as Sarkis said, you'll learn something about the phenotype and interventions that you can then maybe use for a clinical trial. Another possibility is you use in vitro work like stem cells. You try and find agnostic or gene-targeted approaches. You see if they work in your stem cells. You can make your model. You can try them in your mouse, and you can ultimately do clinical trials there as well. So that's a pretty robust direction the field is going. This is one example of Infilo Mcdermott syndrome, actually, where some deficit we saw in the mice, both in the brain function and in motor abnormalities, suggested a certain approach, and we saw some interesting effects of a specific growth hormone, a growth factor in patients with this disorder, which we've now submitted a follow-on study. However, as good as all that is, we found 250 genes out of 1,000, and many people will not have a mutation. They might have genetic risk, but not a formal mutation. So this approach is only going to get us so far in the autism spectrum. So the other approach would be not out of many, one, or out of many, but e pluribus paucis, which means out of many, few. Can we take 100 genes and come down to just 10 pathways? And instead of thinking about treating each gene mutation independently, we think about treating each of those 10 pathways, depending on the individual, and that individual may not even have a mutation. They might just have a deficit in a pathway that other people with autism have as well, and that would be the point of intervention. So it is this pathway targeted approach. So then the question becomes, how do you get from these rare genetic mutations to actually find common biological pathways? And that is really the subject of the funding. And Matt is going to take it from here. So Matt, are you going to share your own screen, or do you want me to just advance?
You can throw mine up, I think.
Okay.
$2,000 years to develop therapeutics for the existing genes that we've already discovered. And as the rate of gene discovery continues to grow, it's clear that we need new approaches to try to come up with treatments that can work for many of the genes at once. But before we can do that, we need to establish a system that would allow us to study many ASD risk genes at once so we can discover their shared pathways. And we want to do this in human cells. And then using this system, we want to identify FDA-approved drugs that can restore the disrupted pathways. And we're extremely grateful to the Brain Foundation for funding this idea that we have. So autism is a neurodevelopmental disorder, which means it affects the developing brain, one of the most complex organs in the body. The part of the complexity of the brain is its many cell types, some of which are shown here, and all of them thought to play a role in autism. So for each of the ASD risk genes, we really need to understand the function of these genes in each of these brain cell types. Unfortunately, the human brain is inaccessible to us to study these cells directly, but a workaround for this is something called reprogramming that won the Nobel Prize a few years ago, where you can collect blood or skin from individuals and then revert them back to what we call induced pluripotent stem cells. And then in a process called differentiation, we can direct these cells to become any cell type we want, including cell types relevant to ASD. So this is the best system to study human neurons, especially for neurodevelopmental disorders. So the landmark ASC paper that Dr. Buxbaum presented found that many ASD risk genes are involved in gene expression regulation and neuronal communication. And what you can see here is if we look for their expression over the course of brain development in humans, you see that many of them are strongly expressed in the developing brain, which is nice because we can use the induced pluripotent stem cells that I described to generate these cell types in a dish. And we've done that using the iPS cells. So here I'm showing the neural progenitor cells that we've differentiated from the iPS. This is what they look like by bright field. And then we can stain them for various markers of neural progenitor cell identity, which is shown here. So we've generated these, and this will form the basis for our assays to study the function of ASD risk genes because it matches their developmental expression. But the top goal of the project is to study many ASD genes at once so we can compare their effects and find convergent mechanisms. So on this slide, I'll tell you about our plan to study these genes at once. So the foundation of the project is a technology based on Crispr interference. And so here's an example of an ASD risk gene being transcribed into its mRNA. So we can use this Crispr interference technology to direct a protein called dead Cas9 to the start of this gene and thereby block its expression. This mimics a loss of function mutation, which many of the genes that Dr. Buxbaum talked about act through loss of function mechanism. So the critical piece of the CRISPR-Cas9 is this short piece of RNA called a guide RNA that enables us to direct this Cas9 anywhere in the genome. And importantly, this makes it really high throughput because we can generate vast numbers of these guide RNAs targeting each of these individual genes. So now I've described our IPS system and our CRISPR-I technology, and now we're going to put it together to make a platform to study many ASD genes at once using this. So we've generated a guide RNA library targeting 75 of the high confidence ASD risk genes. Another feature of this technology is that when you add it to the cells, we can add it to neural progenitor cells so that each cell gets only one perturbation or one of these ASD risk gene targeted, as shown here by kind of independent colors in each cell. And then we can differentiate these neural progenitor cells into neurons or different types of neurons or any cell type that we think is relevant to ASD, like OPCs, for example, although that's beyond the scope of what we've proposed so far. So now we'll have a pool of these cells, each investigating one ASD risk gene, but studying many of these genes at the same time. So how do we tease out the effects of each of these things? We use another technology called single-cell RNA sequencing, in which we put these individual cells into these tiny droplets and then perform RNA sequencing on each of these separately. So single-cell RNA sequencing allows us to generate the transcriptional signatures of ASD risk genes. So gene expression profiles, they represent a high-dimensional signature of cell activity, health, and identity. So we can use these signatures. So this is an example of a signature for each of the ASD risk genes that we've obtained from single-cell RNA sequencing. So we can use these signatures to see which biological pathways are commonly disrupted by ASD risk genes. And these signatures are going to be in a cell type specific and time point specific manner. So importantly, and the whole reason we're doing this method is because we can now compare many of these genes at once to find patterns shared between these genes. And these patterns represent convergent mechanisms across these genes. So instead of targeting each gene separately, we can now think about targeting groups of genes that act the same way. So I'll get back to that in a second. But I first wanted to show some preliminary data that this method works. So in work that I published last year, I demonstrated this approach looking at 14 ASD risk genes, and I found that 5 of the 14 that I studied, as shown here on the left, had strong overlap of their transcriptional signature, which means they shared downstream target genes. When we investigated which pathways were affected by these genes, we found some gene ontology terms that suggested that neural progenitor cell differentiation and proliferation were affected, which is again why we're using the human IPS neural progenitor cells in the differentiated neurons now to follow up these findings across 75 of the top ASD genes. So going back to our single cell transcriptomes and thinking about convergent targets for therapeutic intervention, one of the emerging ideas for drug discovery is reversing gene signatures. So here's a schematic of that, where you can see here that genes B and D have the same signature. So now what we can do is look for drugs that have the opposite signature in the same cells. And the idea is that these kind of cancel out, so that drug X would, by adding it to cells with these mutations, would actually reverse the signature and push these cells back to a healthy state. And we can do this now because all of the FDA approved drugs have now been tested in iPS neurons and generated signatures for each drug. So all we need to do, using some elegant algorithms developed at Mount Sinai, is to look for these drugs that can reverse the signatures. So this is one of the main goals of our project, and hopefully it will lead to some therapeutic discovery. But we can also ask some other questions in our single cell data. Without getting into too much detail, and for more details, you can refer back to my 2020 paper, but we can also use this data to investigate whether ASD risk genes alter the pace of neuronal differentiation. So our data ends up looking something like this when you plot it in this pseudotime analysis, where you can see that differentiating cells choose their fate. So because we can see these fate choices, we can then ask, do ASD risk genes contribute to alterations in cell fate? So one of the leading theories in autism is a disruption of the balance between excitation and inhibition in neuronal circuitry. And this could simply be from generating too many or too few excitatory or inhibitory neurons. So we would be able to see that in our single cell data. So, and many of the ASD risk genes might act in the same way or in opposite ways, allowing us to come up with separate therapeutic targets for each of these sets of genes. So that's kind of the big picture view of everything that we're working on, or not everything that we're working on, but what we proposed in this Brain Foundation grant. So I'll end there with a view from our lab. And I want to acknowledge the generous support of the Brain Foundation. I'm also supported by the Beatrice and Samuel Seaver Foundation, BBRF, and I also want to thank the ASC, the NIMH, and my team and colleagues. And I'll let Dr. Buxbaum take over again, and I'm happy to answer any questions in the chat. Thank you.
Thank you, Matt. Yes, we're happy to answer questions.
Thank you, Joseph. Thank you, Matt. There are a couple of questions in the chat. It looks like one for each of you. So Joseph, we'll start with the first one, and I'll just read the question. If an environmental factor is driving the rise in numbers of autism, so I guess maybe you can just address that premise before I go on with the question. So if the environment is driving the increase in the numbers of autism, wouldn't it make sense that as time goes on, it would present as if a genetic subpopulation, which is simply reacting to that factor, is more likely to have autism, whereas the true causative agent is the environment? How do genetic studies circumvent this problem?
Yeah, so that's a good question, and there are a bunch of good answers. But first, I think most people in the field would say that the rate of autism increasing is largely at least 30-fold increase simply by change in diagnostic criteria. But genetics actually can handle an environment, and we actually formally model it. There's a famous quote by a British epidemiologist and geneticist who said, if everybody smoked, lung cancer would be a genetic disorder, right? So if there is a consistent environment that, everybody shares, there's no change in the environment, you only see the genetic effect. If there's no change in the genetics, you may only see the environment effect, right? So height is one of the most genetic traits we have, and yet people over the past 200 years, everybody gets taller and taller, right? It's because that there is very little change in the environment and over the course of a study, but there's big changes in environment affecting height over the course of decades and centuries. So we're actually able to do a pretty good job of teasing apart environment versus genetics. And but I would say the most important thing is that those exact things, whether it's people say it's 60% genetic and 40% environment, those are pretty meaningless because depending exactly on what's changing, it could become very different. The important thing is whether there's any genetic component, and if so, can you find the genes, or if there's any environmental component, can you identify them? And that's really the important thing. And whether, some people say it's 50% genetic or some people say it's 80%, in biology, those are the same number. Biology, biologists have what's called physics envy. We're happy if we get the sign right. We don't have anything down to 4 decimal places yet.
A plug for quantitative biology there at the end. I think this next question is for Matt, so thank you so much. What's your opinion on bone marrow stem cell infusion via cerebral spinal fluid?
I think there's been a couple of studies that looked promising. I think the largest one failed to replicate. I think if they're safe, people should. I think they all need to be tested extensively for safety before we can even talk about anything with efficacy for these, just like any drug. But hopefully there's some therapies emerging on the near-term horizon, wherever they may come from. I don't have a specific opinion on that, though.
I will just add that the stem cell work that we talk about is different from stem cell therapeutics, right? In stem cell therapeutics, you're putting in cells into somebody under different circumstances. Here, we're using stem cells to make brain cells in vitro, so we can study brain tissue without actually doing a brain biopsy. So the stem cells here are used because stem cells are able to make other cells, any other cell in the body, ideally, and so you can create human brain cells in vitro in a petri dish, and that's how we use stem cells, which is very different than the clinical application. But as Matt said, there's not been anything very compellingly positive. Most of it negative, and one of the ones that generated some interest a few years ago was just retracted because of significant ethical concerns as well. So I would say we don't have actually any affirmative evidence for stem cell therapeutics in autism.
And one final question, I think this is also for Matt. Can this be tried on individual patient sample? I'm assuming it's the iPSCs, especially say very positive responders from previous trials to validate?
Yeah, that's a nice question. And it's something we're doing in parallel, as Dr. Buxbaum showed in the table. We have several teams, but it takes teams of individuals to study just one of these disorders from one set of patient-derived IPS lines. Whereas my approach, a single person could generate this whole library and study all of the genes at once. So in terms of learning the function of the genes, I would recommend my approach. But yeah, then validating any findings, it would be valuable to have the patient-derived lines as well. And pharma is especially interested in the actual patient-derived samples for their validation. So I think they're nicely complementing systems.
Terrific. Thank you, Matt. I have a couple of questions as well, but in the interest of time, maybe we'll move forward and I'll track you down offline. Thank you, Joseph. Thank you, Matt. So now we'll move to the talk by Stuart Lipkin. Stuart is the Hannah and Eugene Stepp Chair and Professor, as well as co-director of the Neuroscience Translational Center in the Department of Molecular Medicine at the Scripps Research Institute. And the title of Stuart's talk is Potential MEF2 Transcriptional Activator to Improve AST Phenotypes.
MEF2C Association in Europe, many of them are on this teleconference due to the magic of Zoom. So you're truly international today. So I'm going to talk a little bit about, well, first, thanks for having me back. It's always nice to be invited back. Maybe that means that you did something good the first time. So I will catch you up on what I said last year, and then we spend most of the time on this MEF2 transcriptional activator. And I just want to give a shout out to Matt, because I was able to remove half my slides because he gave a lot of the IPS and CRISPR-Cas9 that I was going to talk about. So that's fantastic. I can just show you the results without having to worry about that. Matt actually mentioned this, that excitatory inhibitory imbalance is thought to underlie many of the abnormal phenotypes in ASD. I'm having an issue with my screen here, but I think I fixed it. The transcription factor MEF2C is involved in multiple forms of autism and intellectual disability. And so we hypothesized that boosting its activity might normalize EI imbalance and neurobehavioral abnormalities. And we screened what's called the Scripps Reframe library. The Scripps Reframe library is a unique library that Scripps chemists, Scripps, I am so honored. Scripps has it's been ranked 1 or 2 in the world in chemistry. The chemists think I know some chemistry, so we're all going to be quiet about that. And they put together a library of not only all the FDA-approved drugs, as Matt had mentioned, but all the EMA-approved drugs in Europe and other compounds all over the world that have made it through phase 1 safety trials, even if they're not yet on the market. So the beauty of that approach is that we have all the drugs basically that have been ever tested in human that are safe, and we were able to test those. And the lead candidate I'll show you today has extensive experience in humans for another indication and shows excellent MEF2 activation and corrects EI imbalance. And I'll show you some of that data. Okay, so this is bringing coals to Newcastle. You've heard this in multiple other talks. The latest CDC statistics are now 1 in 44 children. You've also heard Dan Geschwin's name in both Sarkis's talk and Joe's talk. There are multiple slides. Dan is also my colleague, and he looked at, I'll show you the data in a moment, a series of ASD genes and was able to parse those genes into families. And interestingly, the transcription factor MEF2C and MEF2A, a related transcription factor, control many of those genes. Dan then contacted me. I was the discoverer of MEF2C, and with Dan, we've been looking at MEF2C in various neurologic diseases, including autism spectrum disorder with intellectual disability. So our reasoning is that therapies aimed at improving MEF2C-associated ASD phenotypes might potentially affect many other forms of ASD, because we know, not from iPSC data, although we do know, from real human data, that MEF2 controls many of the genes involved in ASD and intellectual disability. So here's some of Dan's data from a paper in Cell, and you can see these 5 hubs of genes that are many of the genes that Joseph showed you in the prior talk. And MEF2C and MEF2A are 2 of a few transcription factors. Transcription factors are proteins that go in and turn on genes. So they control many of the other known autism genes. Now, interestingly, MEF2C itself can be mutated and form a very severe form of autism and intellectual disability. We first discovered this actually in a mouse, and then colleagues subsequently discovered it in humans. And so this is a gene that encompasses the MEF2C region, and now we know specifically of many mutations in MEF2C that cause this. If you look at the UK database and some other databases, it has the possibility of being extremely frequent, in that more frequent than, say, Rett syndrome. Oops. And these children generally have severe autistic behavior. Only a few of them talk, accompanied by epilepsy, periodic tremor, and abnormal EEG. We'll get back to the electrical activity in a minute, and often severe intellectual disability. Although there are a few that are less severely affected that I've seen. I'm also a physician and have seen, it must be nearly 100 of these cases now, and I'm in contact with many of the families. So we have 2 disease models. One you just heard about. We also use human patient skin biopsies, both from patients that we then use CRISPR-Cas9. We take tractable mutations. These are mutations that are not too large that we then can correct. We've also introduced mutations into the normal genome to mimic MF2C. So we have a whole family of these MF2C mutants. Importantly, we've used multiple genetic backgrounds. You must do this to ensure that it's not something in the background that's causing your phenotype. So we have them from multiple families that we've corrected. We also have a MF2C mouse that we published, shown here, and we use that to test our drugs. Although, like Matt Lally, we feel it's really important to do this in a human context in iPS cells. Okay, so let me show you our disease in a dish model of the human MF2C haploinsufficiency syndrome. And so, as Matt just eloquently explained, so I was able to take out many of my slides. We take patients, make human iPSCs, then we can differentiate them into neuroprogenitor cells, into neurons, and multiple other cell types. We've done that not only in two-dimensional cultures, but in these 3D mini brains called cerebral organoids. I'll show you some pictures in a moment. And this just takes you through the stages. These show that we've made NPCs, that is pluripotent cells. We then can take them through the various stages to make neurons. They start to look like neurons, but importantly, they stain to show their neurons. They have the electrophysiological characteristics of neurons. We also have done single-cell RNA-seq, and I'll tell you about that in a minute. So I think the most important finding from this, which should be appearing shortly in Science Translational Medicine, is that the MF2C ASDs make a lot more astrocytic cells and fewer neurons. So here, MAP2 is a marker for neurons, and you can see there's not a lot of green here in the MF2C child's neurons. In wild type, estrogenic wild type, a lot of neurons. And the opposite is true if you look here. GFAP and S100 beta in this context are astrocytes, and you can see more in the MF2C neurons after 6 or 10 weeks in culture, and fewer in the wild type. So you're not making neurons properly, also not making synapses properly. Yeah, so I mentioned we can make these 3D cerebral organoids in a dish. We've actually brought some of these kids in, told them we created a little piece of their brain or part of their brain in a dish. They look like this. Many of them actually have little ventricles, what you have in your brain, as you can see here. They have layering. I won't go through all the different layering we have, but we get them pretty mature. We've kept them for 6 or 9 months. We do this in multiple diseases, Alzheimer's disease, which I'll come back to at the end, actually. But I'll show you the autism studies today. And then single-cell RNA-seq shows multiple cell types, interesting deficits in both excitatory and inhibitory neurons in the MF2C ASD patient samples versus wild type. And others have shown that MF2C is very important in both excitatory neurons and inhibitory neurons. Interesting also in human microglia, although I won't talk about that today. But we think that underlies some of the infections these kids get. Okay, so let's cut to the quick. What do these cerebral organoids do? Well, they manifest EI imbalance, excitatory inhibitory imbalance. They actually have too much excitation. And this is one indication. We have patch clamp recordings. This is calcium imaging. We have multi-electrode arrays where we can record the whole brain like an EEG. I just want to show you calcium imaging. I showed some of this last year, but we have a lot more to show you now. And so you can see this is the normal wild type, and the neurons are signaling to one another. And we can quantify this. I'll show you the quantification in a minute. If you look at a kid, a child, iPS cells, this child happens to have seizures, and whoops, we've actually recreated the seizure in a dish. You can see it pulsating like that. We can quantify that. Not all of them have seizures. Sometimes they just have too much electrical activity. So the EI is balanced in this case toward E. Too much excitation, you can see it here. Last year, I talked about a drug that we're developing called nitrosynapsin, and you can see when we put it on, it calmed things down dramatically. Here it is again, we calm it down, see everything gets much more toward normal. So as I mentioned last year, this is an interesting type of glutamate receptor antagonist called the NMDA receptor. It's based on a drug that I patented actually for Alzheimer's disease. It's now one of the FDA-approved drugs for Alzheimer's disease called memantine. We modified the structure of memantine, and it has a second site of action at a site we call the S-nitrosylation site, more chemistry that had been discovered in my lab some years ago. So this is a dual-acting, very, very potent drug, but very, very safe. Little company in Boston is developing it. Here's an update on that, and then I'll get to the MEF2 activators. So I won't show you again, but I showed you last year, and we published it, that this drug, nitrosynapsin, damps down only the excessive excitation, doesn't affect normal excitation in the brain. It also corrects multiple neurobehavior abnormalities in our MEF2C mice. We've now nearly completed pre-IND studies in preparation for a human clinical trial. We're actively seeking financial support. There's a major philanthropist who's involved in autism who's considering funding the trial, but we'd love to include any of you if you're interested. We are actively raising funds for the trial. We're going to do a phase 1/2 trial at the same time. And we've identified the first site, which will be at Mass General. They've developed a clinical biomarker that reflects this excessive NMDA receptor activity, so we can get an early readout with our biomarker in addition to doing neurobehavior tests. So that's the plan for nitrosynapsin. Let me now turn to this MEF2C approach, which we're very excited about, and which the Brain Foundation and the MEF2C Association have been funding recently in the lab. And so I mentioned that obviously MEF2C haploinsufficiency, where one copy of the gene is missing or has a deletion or is somehow not working, there's one allele, that the other copy of MEF2C that's still there. Perhaps we could overstimulate that and compensate. And then most importantly, because Dan Geschman had shown that MEF2A and C are very important in controlling those hubs of many, many other genes involved in ASD, we reasoned if we could increase MEF2 activity in general, we might be able to turn on those genes and offer some benefit. And so we carried out high throughput screening of the chemical libraries I mentioned at Scripps, not only that Reframe library, but multiple other libraries. We have about 1.5 million compounds. I haven't screened all of them. I don't believe in that mindless screening. We screen for drugs we think are in categories that can work. So we have about 100K screen that we've done. And we've identified 5 drugs that are existing drugs, already been in humans. And we have one candidate, I'm going to call it compound Y today, and I'll show you some data with that compound. So how do we do this? Well, as I mentioned, MEF2C and A, and there's other forms of MEF2, but those are the 2 that we know are involved here. They bind to a piece of DNA and turn on various target genes. What we've done is replace one of those target genes with luciferase reporter, something that bioluminescently has bioluminescence, so we can see it in the screening method. And then we add multiple drugs, and we see if it activates MEF2. And here it's shown in a 384 well plate. We have a luminometer to read that bioluminescence. We can put one drug in each, and we get basically certain drugs turn on MEF2 activity, shown here on the y-axis. We have what's called a very good Z-score. We have a good, anything over 0.5 means you can go and screen your libraries, which we've done. So here's one typical screen of many that we've performed. And this is MEF2 activity. Remember, turning on that luciferase. So we're going to turn on all those genes that are turned off, not only in MEF2C haploinsufficiency, but in many other forms of autism. And you can see the top hit here is this compound Y. We do have other hits, but we're particularly interested in this one because it has very extensive use in people for another indication. And so now we more precisely look at MEF2C activity for the aficionados out there. We we compare it to Ranilla. So we just want to make sure it's not turning all transcription on, but just MEF2C. Ranilla is to monitor background activity, if you will. And we have very good activity at MEF2 sites. So we would turn on MEF2A and C in point of fact. Now we wanted an indication. Remember I said the neurons weren't growing properly and they weren't growing out their neurites and their synapses. So we have multiple ways. I'm just going to show you one of them. We can actually look at that neuronal marker MAP2, which is all over the neurites, and we can quantify that as shown on this plot here. And what you see is there's more MAP2 when we treat the haploinsufficient neuroprogenitor cells that have less MEF2C. And you can see much more MAP2 protein. I won't show you pictures, but also we get many more neurites and synapses when we put the MEF2 activator back into the dish or into a cerebral organoid. Okay, so remember I showed you this picture of a child with seizures, boom, boom, boom. We can actually quantify that aberrant electrical activity, giving you EI imbalance. And that's kind of shown here. So wild type, you do have normal electrical activity. Here we're just looking at those calcium bursts. And you can see in the MEF2 patients, this is patient 1, but as I said, we have half a dozen patients, you get these bursts of calcium activity. Whoops. And when we put on compound Y, we still get the normal activity, no more bursts, and it's quantified here. So we are able to rectify EI imbalance. We're now testing this compound to look at various behavioral phenotypes. So we're really excited about it because it's been in patients. It's interesting, it's not FDA approved. So that also gives us an intellectual property approach to it so we can get some company interested in bringing it forward. So in summary, what I told you about today, and I was able to go quickly because Matt, fantastic job, Matt, in kind of giving a lot of the background. I could skip those slides. So we're looking at MEF2 haploinsufficiency syndrome. And particularly, we're interested in this excitatory and inhibitory imbalance as a biomarker, if you will, of drugs that might improve not only MEF2C haploinsufficiency syndrome, but other forms of autism that have EI imbalance. Now, just amazingly, my close colleague at MIT, Li Wei Tsai, published in Science Translational Medicine last month that MEF2C is also important in various forms of dementia. We had been worried if we played with MEF2C later on after initial development, we actually might do damage rather than help. And I think that's a real worry about genetically interacting with any of these genes that Joe or Matt or I have been talking about. But here we have Li Wei, who's vetted this for us, and she's actually found in the oldest old people who have a resilient Alzheimer's or other forms of dementia, they have more MEF2 activity. And in a mouse model, if you decrease MEF2 activity, you get a form of dementia. Multiple mouse models of dementia have left MEF2C activity. And if you genetically increase MEF2C activity, you can abate the dementia phenotypes, memory, cognition, and multiple neurobehavioral tests that Li Wei and her group did. So we're now testing our drug in Li Wei's dementia models as well. And so what does this mean? Well, we think this EI imbalance, and we recently published a review on this in Annals of Annual Review of Pharmacology, is that at the extremes of life, life is a bell curve. We're all somewhere on that bell curve. At the extremes of life, your nervous system is very, very susceptible to EI imbalance. And it's not just the neurons. As other speakers have mentioned, other cell types, both the myelin-forming cells, the astrocytes, microglia, they're all involved in this EI imbalance in an intricate network. We know that, so is the extracellular matrix. And we think you're vulnerable, and you paradoxically at the extremes of life can develop this EI imbalance. And some of the drugs we use in one disease, say in Alzheimer's, might be amenable to being used in autism to correct the biological pathway that's being disrupted. And I showed you that the MEF2 activator compound Y increases not only MEF2C transcriptional activity, but this biomarker phenotype of EI imbalance. This is a drug that's been in people, has no significant toxicity, and so we're really excited about bringing it forward. So I've had tremendous help. Here are the people that have helped me, other labs as well. Here's Dan Geschwin, Jeff Rosenfeld here at UCSD, Nick Shork, my bioinformaticist. I particularly want to acknowledge and thank Pramila at the Brain Foundation, who had the faith in us doing this at a very early stage of the work. Thank you. And Sarkis and Joe, whoever votes for this, thank you all very, very much. And also I want to acknowledge the MEF2C Association in Europe that I mentioned, which is a fantastic group of families with a tough disease, with MEF2C haploinsufficiency, who've banded together really all over the world to try to help with this kind of work. So this is where the work is done. It's a beautiful building right opposite the ocean. I better not talk about this. I know the weather back east isn't good right now. And you're all welcome to visit after COVID, please. And I'm glad to take any questions. Thank you.
Thank you, Stuart. That was really enlightening, and I would encourage people to ask their questions in the Q&A. There's one question, maybe I'll start, and Matt, feel free to jump in as well, but it's more of a general question on the iPSCs and how well they can model the diversity of neurons found in the brain, human, animal, what have you. I guess that's the first part of it. And secondly, to what degree does this correlation or lack of correlation with the diverse neurons, whether it's at the expression level, the activity level, the neurotransmitter level, to what degree does an intervention like a drug or a Crispr intervention inform us about what neuronal subpopulations may be relevant in the human brain?
Yeah, so let me start with that, and then we can get Matt to chime in too, if you don't mind. That'd be really good. Yeah, you're right. Look, we have a developmental model in a dish. Luckily, this is a developmental disease, so it's not that we're trying to model a very old disease. Now, my colleague, Arnie Kriegstein, who has worked on development in iPS cells for a number of years, has wrote a series of papers and also editorials, if you will, about this question. Can you model in a dish a human disease? I mean, you're in a dish. So there's several issues here. One is the cells don't develop totally normally, even in the brain organoid model. They develop more slowly. And, you know, I think particularly this is important for neurodegenerative diseases. You're 65 or 70 years old, not like autism when you get a disease. But interestingly, in the dish, they're under a certain amount of stress. And that seems to speed up the development. So even Arnold has agreed with us that we can mimic many of the features of the disease. But this is why having a biomarker of the disease that mimics the phenotype, like EI imbalance, is so critical here. What I'm telling you is I can, in a dish, mimic a finding that I found in mice and others have found in EEG in patients. And I can use that biomarker to use my drug discovery. So like Matt, we're also doing something called Cmaps Link 1000, where we have thousands of drugs that fix transcriptional activities, and we can screen drugs that way. You know, I think that's fine. Matt, I actually didn't know that it had also been done in human neural progenitor cells. I may write to you to get some more data on that, because the database I have is not just for human neural progenitor cells. Now, let me address the single cell RNA-seq data. We do see a diverse number of different cells in our cerebral organoids. Okay, now I can't tell you we have every type of neuron, but we have looked against adult databases, and we have, and that's actually how we classify the cells. We look at a transcriptome signature, if you will, of, say, an excitatory neuron in the striatum. And we can match those, and we do see a vast repertoire of cells, but I know we don't see all the cells in the cerebral organoid, but we do see many of them. And so we're encouraged that because we have that phenotype, that electrical phenotype that we're very good at monitoring, we think we have some concordance, if you will, on the disease.
Sure, thanks. A couple more quick questions, I think. Is a planned trial for compound Y in patients with MEF2C mutations or in a different subset of ASD?
Yeah, so that's great. There is no, although we're working with colleagues to get this, we really don't have what they have for Rett syndrome, which is a natural history study of MEF2C, which would really facilitate our doing a clinical trial. While we'd like to do that, I think all of these drugs, and we've met with the FDA and discussed this ad nauseum, we're probably going to be doing it in another subset of ASD patients that are a little more tractable. Yeah, so both for nitrosynapsin and the MEF2 activator drugs. That's the plan right now, simply because the FDA is driving us that way, but we hope in the future we absolutely want to treat those children. Look, all of our children need help with this, and we're working hard on that, as we all are at the symposium.
Yeah, I could imagine having a pre-screen based on a narrow genetic subpopulation, which is, you know, make recruitment so challenging that it would be, you know, very difficult to even run the trial, right? I'm just wondering, maybe just dovetailing off that question, because MEF2C controls so many other genes, are there other, you know, either genetic or maybe physiological parameters one can use to enrich for a population that may respond to the drug?
There is, as I mentioned, the Mass General group has developed a biomarker that you know, they'll be publishing and talking about soon, and it actually reflects the phenotype I showed you. And so we're very, very excited about that biomarker, because, you know, when you have a little company taking a drug forward, and I've done this several times now with memantine and other drugs, you have to kill a drug. You have to, this is the trick of a little company. You don't make a drug, you kill a drug, so you get to the right drug eventually. So having a biomarker for an early readout in a study is absolutely critical if it's a reliable biomarker. And we think we have one.
Joseph, you have a question?
Yeah, I was just wondering about, you know, some of the, at least the nitrosynapsin and even this new compound Y, they seem to be working on, as you said, excess electrical activity. What about trying it in just epilepsies? Intractable epilepsies, for example.
Yeah, well, that will be one of the readouts. Many of these kids have epilepsy, so that is one of the endpoints we've discussed with the FDA.
Thank you.
Stuart,
Great talk.
One more question in the Q&A. Does nitrosynapsin activate mTOR?
Well, downstream it may. What it does is, I haven't looked at that specifically, but we know that it, well, we partly have looked at that. So we've tested it in a tuberous sclerosis model, and it's published. And I'm not going to be able to conjure up that immunoblot in my head right now. We've looked at the pathway, and it corrects the pathway. It does interact with the mTOR pathway. So the answer is yes. And we think that's because it's, in part, many of these genes are neuronal activated genes. And we know that MEF2 damps down this excessive electrical activity. Interestingly, it spares normal synaptic activity. That's why it and memantine that I developed previously are so well tolerated, as opposed to many other drugs that have tried to interact with the NMDA receptor.
Sure. And Joseph, is that a legacy hand that's raised, or do you have another question?
That's great. Sorry, let's remember that one.
We'll move on. Thank you so much, Stuart.
Thank you all. Thanks for having me.
Absolutely. To the next talk by Kazu Takahashi.
Okay. Can you hear me? Hello?
Yes, we can.
Oh,
Yes.
okay. Okay. So, first of all, thank you so much for having me and bring me here to give to share my story about mannose binding lectin MBL. So I wasn't sure what to do, but so I decided to give kind of broad sort of background what's MBL and my thinking about MBL at this stage. So at this point, MBL to me, it's multifunctional. I hope I can convince you what I'm telling you is true. And MBL is like Sentinel, so providing constant surveillance through, and also kind of house cleaning, and also through coagulation. I'd like to call it bionic glue. And lastly, it is a disease modifier. So as long as you don't have anything, you may not feel it, or you wouldn't know till you look for it. But if you have underlying problem or some infection, then MBL deficiency in particular would make you feel something wrong. Now, I'd like to start with a big picture, at least to me. So thanks to host defense system, we have innate immunity, which kicks in seconds or minutes. Really, after our body is exposed to danger, could be just injury or invading pathogen, and provide local containment. And one of this system is called the pattern recognition molecule, which MBL belongs to. And if we now this sorry. Oops. Sorry, so oops. Sorry, I'm messing about. Okay. Now, innate immunity has linked to acquired immunity. Now, which takes at least a week lag time, but provide systemic and target specific protection, often quite for lifetime, since it has memory component in it. Now, what is MBL? It belongs to the collectin family, and as I mentioned, pattern recognition molecule of innate immunity. By itself, functions as opsonin, which facilitates phagocytosis by so-called professional phagocytes like a neutrophil and macrophage. And also in cells, has served as a post-translational glycosylation. So located border of ER and Golgi, which was actually done by the other research group. And also Mbl in complexes with a mass, Mbl associated stem proteases activate the lectin complement pathway and also function like thrombin. Now, I mentioned innate immunity target is broad. So when we started Mbl target, we really thought only pathogens. So bacteria, fungus, and virus. But we now know Mbl can recognize much broader sugars, and ourselves, like once it's injured or transformed like a cancer, Mbl can recognize also some enzymes and amyloid beta, like a Alzheimer protein. Now, as a function, by itself can serve as opsony, so increase phagocytosis by neutrophils and macrophages of Mbl target. And in a complex with MASP1, MASP3, so Mbl functional Mbl is hexamer. So then this complex activates the lectin pathway. So halfway through alternate pathway as well as classical pathway. Now, our contribution to this was this MASP1 Mbl complex can function like thrombin, meaning transform from fibrinogen, soluble form to fibrin clot. And the Mbl is primarily synthesized in the liver, but some small portion expressed in small intestinal mucosal epithelial cells. And I personally believe it should influence on the microbiome. We heard earlier this morning as well as yesterday. And even though Mbl is synthesized in the liver, most circulates in the blood and seeps into tissues. So far, Mbl has been identified in CSF and alveolar lavage and even in the skin. Now, I'd like to talk about the molecular structure of Mbl. So starting from 5', it's so-called system-rich domain, which we think, well, actually it is, to bundle monomer of Mbl, and followed by collagen, long collagen region. Hence, it is called the collecting. And CRD, carbohydrate recognition domain, which is primary target recognition domain. And Mbl has 4 exons, and of which first exon, towards 3' of first exon, there are non-SNP, they're called D, B, or C aleph. So if you have any of these, Mbl secretion is reduced. And in addition to this, at least 3, actually we know more, 5' open reading region as well as the 3'. And because of these alleles, we have so many haplotypes. This ended up in broad range of plasma Mbl levels from undetectable to, we've seen 20 microgram per ml, which is a lot. But average, if you are wild type for both alleles, 2 microgram per ml. And if you have one heterozygous or homozygous for those amino acid substituting mutation, less than 1 microgram per ml. Now, this is the EM. We never published, but just so that I think you can have ideas. So that we usually call it like a bunch of tulips. It's a hexamer, and probably this one's much easier. You can appreciate the 3 of them. So when we started Mbl, I've been working on this couple of decades, and only interest was infection. So the first report was the children who have recurrent infection had low or undetectable Mbl. And soon afterwards, the life expectancy of cystic fibrosis patients was shorter in Mbl deficient people. On the other hand, these Mbl deficient people are better against intracellular pathogen. At the time, we knew only 12 TB. So because of this clinical notion, we generated Mdl knockout mice. Now I have to mention gene compared to mouse and human. So mouse, they have two protein derived from totally different chromosomes, MBL-A and MBL-C. MBL-A is acute phase protein. So, and while MBL-C is consistently expressed. So when it's acute phase protein, so the naive mice, MBL-A is not really much, but once like LPS challenge, then MBL-A shoots up. Now compared to this, human, we have only one protein, which is genetically homologous to MBL-C. So often some people might describe MBL-C, human MBL-C. But, and if we compare the protein level, actually 40–50% homology between these three human, mouse, A, and C. So when I'll give you phenotypes, I'll talk about Mdl knockout phenotypes. So Mdl knockout mice are actually double knockout of MBL-A and MBL-C. So we made single knockout and cross to make double knockout, so MBL no mice. So I'd like to mention, so there are, sounds very similar molecules out there now. First of it, it's LMAN1, which is also called ELG-IC53 or mannose receptor, which we also worked on. I'm not going to talk anything about this, but both these sound clearly similar molecules are on membrane protein. And so when we made Mbl no mice, mice were healthy and fertile. So this was really great for us. And also we didn't see any obvious phenotype even at the histological level. And we made sure there's no transcript, no protein, and lectin complement pathway was undetectable. Most of the talk I'm going to share are already expressed. So if you would like to see detail, please read the original. Okay, and so first of phenotypes we expected, yes, Mbl mice are susceptible to the infection and impaired acute inflammatory responses. So I would like to share two cases. One, Staph aureus blood-borne infection. So we inoculated mice with Staph intravenously, and red column for MBL null and gray for wild type. So this data is bacterial titer 2 hours post inoculation. So you can see significant, at least 1 magnitude more bacterial titer in MBL knockout mice compared to wild type mice. And at this 2 hours, we also looked at cytokine TNF alpha IL-6. Both were diminished in MBL null mice. So, and second infection example was a viral infection, influenza A virus infection, and is the primary lung infection. And in this data, MBL null mice are in closed bar and wild type in open bars. So day 1 post infection, still MBL knockout mice lungs could not clear a virus, while wild type almost kind of undetectable. And we also looked at, just part of inflammatory response, we looked at white blood cells and percent of neutrophils. You can see response, these white cell numbers and neutrophil populations in Mbl lungs were different from wild type. And I was drawn to imaging all the time. And at this time, I had only FDG accessible. But I did chronological imaging post-infection day 1, day 4, and day 8. Left panel showing wild type mice and the right panel showing Mbl null mice. So by day 8, there was almost no signal in the wild type mice lungs, but you can see some activity, FDG uptake activity in Mbl null mice. So supporting again, Mbl null mice have aberrant response to inflammatory response. Now, based on the Mbl pathogen, broad pathogen recognition, there is a blood cleansing. It's like a dialysis, but the Mbl cartridge is incorporated. So the idea is to clean pathogen from blood stream. It's early stage of clinical trial. And now from here, I'd like to share with you the phenotype we didn't expect, because we didn't really expect much actually. But surprisingly, first of all, Mbl null mice responded greatly to polysaccharide vaccine. And so we had a collaborator at the time working on groupie strep type 3 polysaccharide, which was conjugated tetanus toxoid. And altogether, we immunized 3 times, 20 days apart. And this IgG titers are measured at day 60. And this data shows you groupie strep polysaccharide 3 specific IgG. We also measured just the whole total IgG, IgM. We didn't see a difference between wild type and Mbl. So I'd like you to focus on between wild type and Mbl, and increased IgG isotype was IgG1. And then this increase was in part increased total peritoneal cell number. So peritoneum, that's where we immunized. We used to immunize. And B1 cell was significantly increased in Mbl null mice. And so the next phenotype was coagulopathy. So Mbl null mice had a prolonged bleeding time. And now in mice, you can do kind of simple experiment. So you cut 2 millimeter tip of a tail and put in 37 warm PBS and time till you stop seeing the blood is coming into PBS. So most wild type stops less than 1 minute. While Mbl null mice, many of them didn't stop even 15 minutes. And at this time, we wanted to make sure, the other coagulation factors are not altered in these mice. And as far as we looked at, like platelet numbers and platelet functions were comparable to wild type. And more so when we pretreated these Mbl null mice with recombinant human Mbl, the phenotype went back. So we could say this prolonged bleeding time was due to lack of Mbl. And C3, now by the time we had a mice, people start saying, oh, Mbl is a complement protein, but this result shows you this prolonged bleeding time was C3 independent. Now, as I mentioned earlier, Mbl forms a complex with MASP1. And so we subjected to the same experiment, MASP1 knockout, MASP1 null, and MASP2 null. I'm saying MASP1 null, but actually, so what MASP1 null is MASP1 3 double null, and MASP2 null is MASP2 and SMAP null. So as you can see, MASP1 null mice had prolonged bleeding time. but not MASP2. So we conclude that this prolonged bleeding time coagulopathy was due to lack of Mbl and or MASP1. And this work was done by Peter Gall's group. So MASP is directly linked to pro-inflammatory pathway. So here is the Mbl target and Mbl MASP complex and PAR, protease activated receptor. There are 4 of them nowadays. And so what happened is when this complex binds to the target, Mbl target, MASP can activate PAR and seems to like PAR2. By the way, PAR1 was also known as thrombin receptor. And this PAR activation actually directly linked to pro-inflammatory cytokine signaling pathway. So this is a link between Mbl being acute phase and at the molecular level. And the next was bound phenotype. So we used dry burn, so we burned the back of mice. And in the wild type, what happened is ESCA or the burnt skin slough off overnight. But it was not, oh, this is a picture. So it's kind of expensive, but I was told that mice don't really feel pain. Well, any case, so the wild type ESCA shed off, so you can see muscle, back muscle, while Mbl no mice didn't. And so most 21 out of 23 wild type lost ESCA like this, but only 3 out of 18. And in other words, 15 out of 18 had kind of looking intact skin. And once again, this phenotype was reversed or corrected by adding pretreating Mbl no mice with recombinant human Mbl. And again, C3 complement 3 or complement 4, complement 1Q knockout mice were more like a comparable to white type mice. And we wanted to see what's going on. So we did histological analysis and kind of adjacent skin to the burn area. And one of which we did was to look at MMP. We used fluorogenic substrate for collagenase and gelatinase. You can see this green area. Those are MMP active, while we didn't really see much in Mbl no mice. And more surprising was Mbl no mice, the skin got almost like a 2 or 3 times thicker overnight. And we didn't really do anything further. Now, many of these experiments were done using anywhere between 8 and 12 weeks mice. Now, we wanted to know if there's any age-related issue. So we aged them average like 20 months. And what we saw was nausea, non-alcohol liver steatohepatitis, or simply fatty liver. And when we sent organs to histology, we sent out. And one of the things they noticed was fatty change, meaning the color of liver was actually different. And so we looked at, we did oil red or staining, which stains lipid. So this is wild type and 20 months old. You can see here and there this orange color that means the lipid droplets. In contrast, we saw 2 types of lipid deposit, in small deposits all over, or a huge lipid oil red or staining. And we consulted pathology, and they said they see similar pattern in human as well. And we also looked at fatty acid analysis, and 20 out of 23, I think 16 and 18, we had only 3 each, white hat and Mdl knockout mice, but 16 and 18 change seems to be higher in Mdl knockout. Now, we never published it. We couldn't find any collaborator nearby. So, but we noticed the Mdl mice are tame and very sensitive to any stress. I know this characteristic is the most relevant to this meeting, but we haven't really done anything. The one thing maybe close, or at least something happened in the brain is one collaborator at the time was doing traumatic brain injury in the mice. So Mdl mice had a slow recovery. So, this was just only in vitro. Lastly, this probably relate to brain house cleaning. So this was just like an ELISA type simple binding asset, but amyloid beta 40, 42, and this is just as a kind of control for the experiment. And red crossed bars shows you their binding at 100%. And interestingly, the sulfate sugars inhibited the binding to A beta. And based on this results, even though people didn't really think this system-rich domain as a binding function, it's most likely this system-rich domain, not just bundle make form oligomer, but functioning as a binding domain. And so I'd like to believe that Mbl probably working to maintain balance of amyloid beta. Now we know our brain seems to need amyloid beta to brain health, I believe. So I think somehow Mbl participate sort of orchestrating whole innate immune or homeostasis somehow. And so the summary of Mbl effects. So the Mbl status is unnoticeable in general. You wouldn't know, you don't feel it, but it might become an issue if you have underlying conditions and some infections. And it can be 3 ways. Of course, you know, just healthy, nothing, but too little or even too much seems to be not really good. And when I started, I showed you earlier, we had only a few clinical observations, but now the list has been expanding, and autoimmune diseases and also Crohn's disease and some neuronal programs. And also when we studied, only deficiency was kind of big deal, but now we know too much is not really good either. Now, I'd like to touch a bit what I'm primarily involved with is the development of PET tracer. To me, seeing is believing, and but pain-free kind of slow development for PET tracer actually. Now, one of which successful is tracer is developed by Dr. Brugaloras, and it's on a phase 1 clinical trial, which is 3F4AP. It's a derivative of MS drug for amylopyridine. So this is a cartoon of a neuron. Neuron is covered or protected by myelin sheath. But if there is any damage, including MS, could be stroke or even Alzheimer, this myelin sheath is damaged and exposed potassium channel, voltage gated potassium channel became exposed, and this tracer can bind. And so this is one, this is, yeah, published. So the, sorry. So we were lucky, I'm sorry, but monkey had an accidental brain trauma injury at this area. And compared to other sort of commonly used tracer, PBR28 or PIB or FDG, CF4AP picked really more strongly. And so the currently under production studies, these tracers are made by radiochemists and metabolic related receptors and some inflammatory related molecules and also pain. And we hope that the tracer, if we hit the right tracer, we could provide early non-invasive and objective diagnostics and possibly therapy too, depends on what we are doing. And lastly, the acknowledgement, I'm member of the Gordon Center for Medical Imaging, and particularly thank you to George El-Fakri, he's the director, and my collaborators, Pedro Bugarotas and Norman Dean, and many people are sort of helped me to study Mbl project, and Alain Zuckertz, who cloned Mbl both human and mouse. If I didn't meet him, I would not be working on Mbl, and Mike Super and so forth. Thank you very much.
And thank you, Kazu. We are short on time, not your fault, because you also inherited a little bit of a delay there as well. But what I think we should do is probably move to break until 11:25. So it's a short 7-minute break. I think we all need a little bit time to stretch our legs. But I would encourage everyone to put your questions for Kazu in the Q&A. I know I have a question, which I'll enter